old fossils. There's one outside of Jordan Hall at Indiana University that is an old lithophite. And you can see, if you know what you're doing, you can actually see where the sporangia are on the outside of the fossils still, in these little axils of the microfills. Except that instead of it being this big, it's this huge 4-foot diameter thing. So how did this diversity arise? So this is a sort of fundamental question in evolutionary biology and understanding biodiversity. When we think of diversity, we often think of something like this and how species rise, sort of this Ernst-Mayr view of allopatric speciation. Things are the same population, maybe a river forms and these beetles in this little cartoon separate, and over time differences accumulate and those differences become reproductive barriers and you've got 2 new species. But botanists, that's great. We think about other stuff. So here's our bunny chicken. This is our hybrid. This is our hybrid speciation. So Edgar Anderson, for example, was a classical botanist who thought a lot about hybridization and its impacts and how the conditions in which hybrid speciation happens. Stebbins, Ledrick Stebbins, was another contemporary of his that thought all about polyploidy and polyploid speciation, which is what we're going to talk about tonight. So polyploidy is speciation by genome doubling, and you've encountered this all through your life without realizing it. One of the sort of famous examples of this is called a triangle of view, which was devised by, sort of worked out by a Korean botanist, James Chun. And these are the triangle of brassica. So the things you're most familiar with, brassica oleracea and brassica rapa. So these are things like cabbage, broccoli, cauliflower. This is turnips and bok choy and spicy mustard seeds. They hybridized at some time, maybe we think about 8,000 years ago, based on our analyses. And that hybrid individual, its genome doubled, and it gave rise to Brassica navis. How many of you have used canola oil in the last year? Right? There you go. That's navis. So Brassica navis is most commonly encountered as canola oil, but there's a whole bunch of other vegetables as well that you've likely had. And what happens here is that when this genome doubling happens, it establishes sort of the chromosomes from each of the species are able to pair with each other, and it restores fertility to these sterile hybrids. But it can't cross back and forth with these other parents very easily because of the imbalance in the number of chromosomes. And so it's a form of instant speciation, really, or a very abrupt speciation. And it's fascinated botanists for decades because it's everywhere when you start looking for it. And we've been able to track it for decades by counting chromosomes, since the 1800s, actually. So if you're wearing cotton today, it's an allotetraploid. And if you enjoyed peanuts, if you've enjoyed spearmint, if you like this big trout, those are all polyploids. If you enjoy wine or beer, French fries, whatever, almost everything we consume or touch is a recent polyploid, and pretty much everything is an ancient polyploid, as I'll show you later in my talk. But outside, if we go outside and look at the frequency of polyploidy in the landscape, about 1 out of 3 plants are recent polyploid species. So that means that if we walk through the garden tonight, 1 out of every 3 plants doesn't have the sort of normal textbook diploid genetics. And it has 4 sets of chromosomes, or 6 sets of chromosomes, or 8 sets of chromosomes. And it behaves in slightly different ways, and I'll show you some of these things here in a second. But just keep in mind that the sort of textbook rules that we think of that apply to most things don't necessarily apply to those plants. And a disproportionate amount of our economically important plants, our agricultural plants, for example, have those kinds of genetics. This is why it's been so challenging, for example, to do crop improvement in things like wheat. It's not simple. It's got 6 chromosomes, and they're in 3 different sets, and they don't really know how it all came together. And for that reason, it's been very difficult to just do crop improvement, do breeding in some of these plants. And that's why they're recently, also when they published the recent wheat genome, which has been sequenced a few times now, it's getting better each time. How's that? There was a recent big press release back in the summer, or maybe a few weeks ago, I can't remember now. But that was such a big deal because it's such a challenging genome to deal with. because of all that history of duplication. So why do we care about this, though, other than the fact that it's common, right? That's great, but what else is going on? Well, we've known about it for a long time because of these, polyploidy and these whole genome duplications are associated often with big phenotypic changes. So Hugo de Vries is a famous biologist from the late 1800s who coined the term mutation. And he coined the term mutation describing plants in a genus that he worked with called Enothera, so the primroses, right? And these Enotheras, he was able to make them, cross them, and they would get these progeny that were huge. And he said they have a mutation, right? That's where he coined the term. Some years later, people realized that mutation was actually polyploid, so they had not just a single base pair change, but the whole genome got doubled, right? And there's big phenotypic changes we see in many cases of polyploidy, but not all. So we often see these big shifts in niche or size of plants, and that's one of the reasons that we're really interested in understanding the differential success of polyploid species, is that there is all this sort of really interesting biology that happens there. But from a genetics perspective, we're also interested in because there's a possibility of not only these sort of instantaneous changes that happen when you double the genome. Over time, we might see changes in the genetic variation within these individuals. So one of the sort of things we kind of ponder as evolutionary geneticists is how ploidal level shapes variation and how selection sees mutations, okay? And the easiest way to think about that is if you're a haploid. So you've got one set of each chromosome. So every gene just has one copy, right? There's one version of it. There's no alleles. So alleles are the other versions. So we're diploids. We inherit an allele from our mom and an allele from our dad, right? If you're a haploid, you just have one. So when we think of bacteria, for example, they're haploids, but things like yeast, a lot of those are haploid throughout a lot of their lives, many phases of their life cycle. And what happens in haploids is that whenever a gene is expressed, an allele, whatever that product is, that's what you get. It's not masked at all. There's no other partner there to cover it. So if it's not a good copy, selection sees it. You die, you don't do as well, etc. And so selection can act on that very efficiently, right, in a population. If you're a diploid, though, you've got two, right? So you have two different versions of that gene working at the same time. One of them can maybe not do so much that it doesn't matter because the other copies working just fine, right? So when we think about recessive deleterious diseases in humans, for example, they're often being hidden from selection and they persist in population because there's this other functional copy still. But it doesn't have to be so deleterious that it's lethal. It can just be it's not as good as its other copy in the environment right now, right? But selection can't see those all the time because they're being hidden by that other functional copy. Now, you make polyploids, and you get, this is a tetraploid, so you got 4 copies of each chromosome, okay? You just kind of keep amplifying the ability of these other copies to mask variation at all the other alleles. And it just keeps going up as you increase ploidal levels. So the ability of selection to mask those copies, and you accumulate all this other variation in the background, most of it probably deleterious. It happens at a much faster rate in polyploid individuals relative to diploids and relative to haploids because of this masking process. So this is a sort of a cartoon of what we expect to see happening. But in polyploids, we typically expect to see increased diversity because of this process expect to see increased diversity relative to their diploid relatives. So if you're a diploid, so this is sort of our carboy water dripping version of this. Each of these is a mutation happening, so the starting population, and it drips at a certain rate into the next generation, and it clicks along at a certain mutation rate, and so many things die. But what you see is that in tetraploids, because they have 4 copies of each gene, they essentially scan mutational space quicker, right? And they pick up more mutations, and they pick up new mutations faster than the diploids do, and they have a lot more deaths along the way too, because they're picking up a lot of really bad deleterious mutations as well. But they're increasing diversity, their genetic variation, in their population at any given time. So what we do expect to see though from this sort of thing are more recurrent double-trace mutations than diploids, and a higher rate of molecular evolution and more variation with populations. And so this is sort of, some of this is like the sort of the fuel for diversity when we think about it. Where does it come from? Well, at the end of the day, a lot of this comes from mutation. So people are fascinated and have been fascinated by polyploid species for a long time because of this potential, not only for these sort of instantaneous things that happen when the genome gets doubled due to these changes in phenotype, but because maybe there's sort of this turbo button on mutation and variation in populations of plants. Maybe we can scan that space faster. And so people have imagined for years that polyploid itself may fuel this amazing plant diversity. So Darwin's abominable mystery of the flowering plants, right? They're very young and they're hugely diverse. And they kind of come out of nowhere. Why and how? And for a long time, people have argued that perhaps polyploid explains that. So what I want to do tonight is take you through some of our work on looking at that question. Where and when are these polyploids across genomes? And one of the fun things I want to talk about is how we find not just recent polyploids from where people can count the chromosomes, which involves a lot of skill squashing cells on a glass slide and not breaking it, and showing how you go in your finger, which I can tell you is not fun. So use an eraser. That's, if you ever want to go squash chromosomes, that's my piece of advice. But we don't have to do that these days. We can find these events through genome sequencing and analysis of genes. So I'll tell you about how we do that, but how we see deep events going back hundreds of billions of years in plant genomes and reconstruct these ancient speciation events. And I want to, along the way, I'll hopefully explain some of the scale and analysis of the data that we do. And at the end, if it doesn't seem like it's going too long, I want to tell you about some fun stuff associating those ancient polyploid events with genetic variation in contemporary plants, and how even adaptation going on right now outside is connected to changes that happened in their genomes 20, 30, 50 million years ago. And I think it's a really fun story. So, all right. So one of the cool things about the genome sequencing era is that the plant that they chose to sequence first was Arabidopsis. It has 5 chromosomes. It's 150 megabases, so it's really small. To put that in perspective, the average genome in a plant is somewhere around 3 gigabases. So it's order of magnitude smaller than your typical plant genome, which means it's cheaper, it's easier, it's hypothetically more tractable to sequence. Well, in 2000, when we got the Arabidopsis genome back, the first version of it, people thought, there's a lot of duplication in here, but it's all scrambled up. And it took about a decade to figure all of this out, maybe 6, 7 years. And by the time over the 2000s, we'd sequenced other genomes. Here is Populus, the genome of Populus, and a cartoon of the genome of Vitis vinifera. This is grapevine, for those of you that like wine. This is its genome. This is what its chromosomes look like. And what's amazing about this is that Arabidopsis has this scrambled up looking genome. And so compared to Vitis, where all these chromosomes, and each of these lines is a chromosome, it's incredibly conserved. So the gene order on these chromosomes hasn't changed much over the last 120 million years of divergence between this plant and this plant. And so what you see here are these solid bars. Those are these big duplicated regions. So you can sort of see the dark blue and the dark blue. Now there's some things that have moved around over time. It's a long time. But when you look at this and compare it to Arabidopsis, it's a completely different dynamic happening in those genomes. What's more amazing about this situation, and this is sort of shows you the challenges of this work, is that Populus has the same number of chromosomes as Vitis, right? Yet it had another round of whole genome duplication in its history that Vitis did not have. Okay? Arabidopsis has 5 chromosomes, but it's had 3 other rounds of whole genome duplication since it diverged from Vitis and Populus. So it's really hard to go outside today and look at the numbers of chromosomes and know anything about this duplication history that we see in their genomes. But their genomes reveal it. As you can see here, it's also not easy to see it, right? There's these little tiny bits that are left in these different parts of the genome. So these rates of variation change dramatically across the genome. And this is an example of another plant in the Brassicacea. This is Brassica rapa, the genome sequence. And this is one way we can look at this genome here. Each of these lines on the outside is a chromosome. And all of these lines in the middle go to their partner chromosomes that are homologous or have a lot of sequence similarity on other chromosomes. And if you puzzle it out, it's not just abstract art, but if you mathematically look at this and puzzle it out, you'll see that each of these chromosomes, there are 3 copies of them. And about 15 million years ago, there was a hexaploidization event, meaning that they had a tetraploid, found a diploid, and they made a triploid plant that then doubled, so they had 6 copies of each chromosome. And this is life as a plant evolutionary biologist. It's basically reverse engineering all of these historical changes that happen. And as you'll see, a lot of the fun stuff that we think about when we look outside comes from some of these events. So how do we analyze these? Well, this is our analysis of gene trees. We definitely have Gene Simmons on the outside here, anyway. And so we analyze, so we find these events by looking at not only these sort of patterns of gene order, and this is really kind of a really good way to do it if you can, but this is the sort of, at least until recently, this was the sort of $2 million way of doing it. How's that? You have to sequence and assemble the whole genome, but it can't be in fragments. And so when we normally talk about a whole genome sequence, it's still often in many, many thousands of fragments. So it doesn't look sort of like you've got this nice little piece of chromosome and then this other chromosome all figured out, right? Really, they're all in hundreds or thousands, really thousands of fragments, tens of thousands. And so getting to something that's really high quality like this is rather expensive. So we've only really done it for crops, right? Where there's a big interest in crop improvement and there's sort of an economic purpose behind it. So doing this in non-crop species or non-model species, you have to look at the genes, all right, and the gene trees. And so one way we can do this is to look at the histories of these gene trees and look for signatures of sort of these baby booms, all right? So if you have a whole genome duplication that happened sometime in the history of these genomes, if you don't, like this is sort of like what our genome looks like, there's no obvious duplication event. It looks like this. So these trees there's ongoing gene birth and death, just like outside there's ongoing speciation and extinction in plants or animals around us. Same thing happens to the genes in your genome. There's this ongoing turnover, birth and death of genes. And there's more births and then they die pretty fast. And so you get these demographic curves that look like this. So here on the x-axis we have the divergence of those genes, so those paralogs, the duplicates of themselves, called paralogs. And this is essentially, you can think of this as time. So here on the 0 is recent duplications, and out here are old duplications. And on the y-axis is the number, or the percent of the genomes is the history. And so we see here that there's a lot of recent ones and there's fewer old ones. But when you have a whole genome duplication event, it looks something like this. And what happens is that a whole bunch of genes, most of the genes, for example, these gene families, there will be a duplication. And so instead of it being this sort of random birth and death process, there's this big baby boom, if you want to think of it like that, that's created in the signatures in the gene trees that corresponds to that whole genome duplication. And so even after, this is the safflower, so for those of you who have heard safflower oil, this is the genome of safflower, and the age distribution of the genes. And we can see there's this big whole genome duplication that happened about 55 to 60 million years ago in the history of safflower. For those of you that were here for Rob's talk, this is the same one that also happened in the history of the silver source. So those are also composites in the same family as safflower. These days, this is going to be an arduous task, there's a lot of curation, so as with a lot of stuff, we now use machine learning to infer and classify these events. And this is actually from an undergraduate project here, the people with all the arrows in their heads, Sally Galuska, Jake Wilkins, and Chris Reardon, who really helped push this machine learning tool that we use to infer these events down across the history of not only plants but animals. We've got a whole octopus project going on that the undergrads are really doing. And this is a new approach for inferring these that's very rapid and inaccurate. And we also look at lots of other species trees and other approaches to place these events in the history of life. And I'll just sort of show you some. We've done this in lots of cases. For example, we have gymnosperms. There's genome duplications that have occurred in the history of all conifers. There's genome duplications that happened in Brassicaceae and papaya, etc. They're all over the place. We recently published the first fir genomes this summer with our collaborator, Phi Lee. There are genome duplications there. All right. And we recently had a paper come out in KS back in May looking at the insects. So this is not a plant, but this is something no one thought there would ever be duplications in the history of insects like this. And it turns out that there are. So there are duplications in the ancestry of the Lepidoptera that we infer there's big duplications there, Hymenoptera, Columbella, and a whole bunch of other things. I'm a botanist, so I like to always say I know my knowledge of entomology is Wikipedia based, so I don't want to misstate anything too much. My graduate student knows more about them than I do. But there are many of these whole genome duplications scattered throughout, not just the plant tree of life, but also the insect tree of life. Not as many, but they're there still. And so they're kind of everywhere. We have these glimpses, right? And that's what I kind of wanted to show you with these little flashes. We have these sort of glimpses of it, but our sampling has never been great. You know, we get what we have, just the species that are available. So getting sort of a comprehensive perspective of when and where these things have happened is a sort of a challenge. And so we and collaborators from all over the world started this project, the 1KB project, about, well, it's been almost 8-9 years now. And we sequenced new genomes, essentially, of almost 1,500 species of green plants from across the whole plant phylogeny. So from those green algae, all the way up to the flowering plants. And this is what data existed before our sampling, and this is after. So we had this sort of really big increase in the density of new sequence data generated by the project. And this is, I don't know how many thousands of hours of work going out and collecting plants worldwide, extracting their RNA, sequencing it up, assembling their genomes. I mean, for us, our computers here, our supercomputers here today, spent about 6 months assembling all of these data, where we were the lead, and then we kept crashing into HPC, doing all the gene trees. I'll show you where that steps out a little bit. But I think we got more angry emails. They were going to send those, I think the astronomers were going to send their probes with afters the lasers, because one of my undergraduates, we discovered that injecting 300,000 jobs at once is not cool. And we discovered that 30,000 at one time was also not cool. And IBM got on the phone at that point. We discovered that we could do 3,000 at once if we then waited 30 seconds between injections of 3,000. So we're in some ways limited by what the job handling could do. But we really test the limits of these HPCs here, and probably contribute to climate change in the Southwest and how much computing takes place to do this, unfortunately. But it's a lot of work, and I'll show you sort of what's involved with that here in a minute. And how does that work? Well, I'll show you right now. So how do we do this? Well, the first place it starts with this DNA sequence, right? So we have to get DNA sequence. And we make these big alignments. And today, you know, we sort of live in the golden age here of sequencing. This is a look at how many sequences were on GenBank, deposited on GenBank, which is the place we put all of our sequences. So one of the great things about bioinformatics and genomics is that it's all open source. And so going back to the Margaret Dayhoff, who's one of the founders of the field of bioinformatics, she ruffled a lot of feathers back in the 60s, the early 60s, when she would print up people's protein sequences and things, and then they put them in these giant volumes. You could buy books of the sequences and have, I assume, many shelves full of books of sequences. I've seen these dogs. They're not a small number. These days, we put them all in a large, public computer database called GenBank. And this was founded back in the late 80s, and it grew reasonably well. So this is the curve of sequences deposited in GenBank from the founding to 1999. And this seems like a lot of data. It's like a nice big curve. But this here is how many base pairs are in the human genome. So sort of big data, but then you realize that whenever they put the human genome on there, they doubled that number in the late 90s, right? So there's a lot of, so it's been growing. So if you look at the next decade, it looks like this. So that whole first decade is not even off the line, all right? And by 2008, we were up here. So an order of magnitude more of DNA being deposited on GenBank. This is today, all right? So all of the first 20 years, those first have been part of this line. And here we are on our way up. Now, today we have, this scale, you can't really see it there, but this is a zettabyte, and this is an exabyte, for example. So we're talking orders of magnitude now on this y-axis. So it's predicted by 2025, most of the data that humans will generate will be sequence data. It'll surpass all other types of big data that we have to handle. And we're not the NSA, we don't have a giant facility in Utah that handles all of this data. It's sort of all around. So my laptop there has some new genomes sitting on it. I think most people are like this. We don't really know what this curve looks like, to be honest, because there's so many sequence genomes just laying around, right? They haven't been published yet or deposited yet. But at least in the public databases, it's just taking off. And there are sort of three projections. So the blue line is if it follows Moore's law. Well, you'll notice that it hasn't really followed Moore's law for a long time. So the rate of growth of computer technology, right, where it would double roughly 18 months. The yellow line, I think, is doubling every 12 months, which is what some folks think it might do. Illumina projects, I believe, this red line where it doubles about every 7 months. And we've certainly, that's the yellow line. And the red line, excuse me, is what it's actually been doing. I don't know, I actually think it's probably is going to keep following that red line. I don't see any reason why not. We routinely now just generate sequence data. And it's become so commonplace that it's not we don't think twice about it. And it's no longer the bottleneck in most experiments or analysis. And I think that this line will keep going up, especially thinking about as sequencing enters a clinical era, everybody will get their data sequenced at some point, most likely. It's just going to keep skyrocketing. There's a lot of different technologies to use. These things are all typically sort of large devices. Illumina you might have heard of. That's the sort of the most common tool that we use to do this kind of sequencing. But things like Roche and Ion Torrent were really cool. Roche 454 and Ion Torrent personal genome machine, the PGM. They were around for a while, but they lost out to Illumina. How's that? But what's really crazy about this and what's driving this kind of increase is that this Illumina HiSeq system, the HiSeq X, could generate in 3 days all of the data that was on GenBank in 2008, right? And so the amount of storage that we have and just managing this is a lot of time. It takes a lot of time and effort. I have about 46 terabytes of sequence data compressed sitting on UA servers, and I'm contemplating, do I delete things or buy more? Buy more storage space. And that's the life. And it's getting even harder. There's even bigger things now. So some of these things like PacBio do really long sequencing. So they sequence really big fragments of DNA. And then new cool stuff is nanopore. And so how many of you heard of nanopore sequencing? Yeah, nanopore though is something you'll probably encounter sooner rather than later in some place. Nanopore sequences using a solid state approach, and it sort of shoots the DNA through these little nanopores and can detect the difference between ATs and GCs that way. And it can sequence really long. So there's a Twitter account that looks at how people sort of post there's a competition to see who can sequence the longest DNA sequence. So you can sequence now whole single chromosomes of yeast, basically. And the limit to sequencing a nanopore in terms of length seems the practical limit of getting DNA out of a cell without breaking it up, which is not an easy task. For these old approaches, like Illumina, which is what we kind of think of as filler, it doesn't matter because the reads are only 200 base pairs long. But when you're sequencing entire chromosomes, it's a whole different game. And I just like to always put this up here because people always think that this is going to be somehow extravagant or expensive, right? To think about sequencing a human genome, for example. A human genome is about 3 gigabases, so that's the size of your average plant genome outside. But doing sequencing a human genome now costs less than braces. So anybody who's paid for braces for their kid or for themselves, well, today you can sequence your whole family's genomes for that same cost. I always like to point out that if you have a very expensive Mercedes SUV, sequencing human genome may cost less than a monthly payment or a couple months of payment, for example. Certainly less than the annual cost of a smartphone now, and less than things like really any smartphone plan and tablet or something. People buy these things routinely on a regular basis, but it costs less than to sequence a genome than it does to. This is a nanopore sequencer. I just like to show this off. This is a picture I took last year. We have these in the lab. They don't require a facility. So look, these people are in lab coats and standing around with gigantic machines on floors that are, they're all gyroscopic and they're oil filled so that it doesn't jiggle around when it sequences. These things you just plug into your USB and go, all right? And it's powered completely by the USB. People take these all over the world in the field and sequence away. And how do they sort of make a sequencing center out of it? Well, they have a giant box full of USB connections, and all they do is plug in 50 of these things. And it goes and you plug in another 50 the next time. And you get even little lab prep tools. All right, so that's sort of how this goes, and I just want to give you a perspective that this is, the future of sort of biodiversity is really going to be tearing apart sort of genomes and pulling them back together again, and it's getting to be a very straightforward task these days. But in this project, we sequenced 1,500 species of green plants. This is the largest analysis to date of this sort of work. But how do we analyze that scale of data? What do we do with it, right? How do we process all of that? Well, as I said before, we got to construct a gene tree for each of these families, all right? And just to give you a sort of idea about how complex of a task this is, I want to just sort of take you through the basics of how this works from an algorithmic perspective. So if we have 4 species, there are 3 possible unrooted trees, okay? And we can just draw all of them. All right, so we got 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4. And we would sort of figure out which tree is best by looking at the actual DNA sequences. So here's our 3 base pairs. And at each of these positions, we got TTA, GAGA, CCGG. And the way that we would do this is use some kind of a model. In this case, this is what's known as the GTR family of models, the general time reversible models. Now, if you can see this, it shows up very well. We've been adding parameters over the last 30 years, basically, from the first model, which just let us estimate 2 of those parameters between the difference, the transition between an A, C, T, or a G. And as computing power increased, we added parameters. These days, we just do all of them. So there's all these possible different changes. So the probability that an A became a C, the probability that a C turned into a T, the probability that a G or a C turned into each other, right? So you have all these possible transitions and transversions that can happen in a matrix, okay? And we can calculate all those probabilities now. And what we do then is that we evaluate the score using that model for these different sets of relationships, right? Whether a T had to become an A then a T then, or a T had to become an A then a G, or just T to an A once, right? So you can sort of imagine the different possibilities and combinations that these relationships impose. And we do this as a sort of a likelihood approach, thinking of it in this way. So we would calculate the score for each of these. And for 3 gene trees, there's not really a giant peak. But if this was a really big data set, you have many, many scores. And it makes these landscapes in the numbers, right? And so we talk about finding the peak likelihood, all right? And so the tree that's at the peak of that likelihood score is the one you want. And that would be your tree that would explain those relationships the best according to that model that you picked. But that's hard to do. So tree space is vast. For 3 species, there are 3 possible, for 3 rooted trees at least, there are 3 possible trees. For 5, there are 105 trees. For 50 species, right? There are more possible trees than all atoms in the universe. All right. So it scales really great as a computing problem. So what about 1,500 species, right? It's really weird. So to put this in perspective, for 500 species, it's 1 times 10 to the 1280th possible trees. All right, so we're all gonna be sitting here, the sun's gonna burn out, and a whole bunch of other stuff's gonna happen before we ever compute all possible trees and ask what's their likelihood for 500 trees, let alone 1,500 trees, or 2,000 trees, 2,000 species, right? It's just a very difficult problem. So how do we solve this? Well, one way we do it is we sort of cheat, and this is we explore tree space. This is the old school way, right? And what we do is we just swap things around. So we can't actually look at everything. So we use a heuristic branch and bound approach. We just swap the thing, test it, is it better or worse? If it's better, then we keep that class of things and swap another big chunk. We can't move all of them around to all the different options. The danger there is that you get stuck be worried about getting stuck on a little island of maximum of a good score. But really over there is the big peak, right? And we just don't know sometimes. That's the way it works. And you try to get through that as efficiently as possible. And as I say, we hope we climb to the top of Likelihood Mountain. But as Mike Sanderson, who's a colleague of mine at EEB, has shown, getting to the top of Likelihood Mountain can be tricky, even if you're on the right mountain. How's that? Because there are these terraces of trees that are have the same exact score. And this gets worse the more data you have. So you keep changing things, but the score doesn't improve or get worse, and you just think, okay, am I at the top of some flat mountain, right? Or am I actually just stuck on a terrace? Not an easy problem to solve. The mathematics of this are pretty intense, and this is a whole class of problems, but it'd be hard problems. And if you solve one of them, you solve a lot of things. I don't know that we're going to solve this anytime soon, but we can always hope. And when we do this for a typical sort of group of plants like Selaginella here, this is a phylogeny made for the Sonora Desert Selaginella. What we can do, we can kind of manually do this and check things. But when we're doing this for 1,500 species with 8,000 genes each in the analysis, it's really difficult to do that manual curation. And so what we need, and this is what the sort of big tree looks like. And so what we do, we have the scale of data. It's a next generation approach. Insert bad Star Trek joke. And so in this sort of approach, what we do is we iteratively go back and forth between doing these sequence alignments and the tree itself and see how things update. And this is a divide and conquer algorithm. And what we do is we decompose the tree into these little sub-problems, work on those little problems, put it back together again, and keep doing this. And we get to the end of that tree, and we say, is it better or worse? If it's not better than the starting tree, we keep it. If it is better, we accept it and do this iterative process again. This is, right now, actually produces the best trees possible, and actually gets really close to sort of what we think of as the gold standard of human curation of being able to get this. Because again, we have this whole heuristic problem of getting through all this data in a reasonable amount of time. And so, this is our big team effort here. And in order to produce this tree for the 1KP data and find all these polyclades on it, we have this giant scroll, which I'm ready to unroll. Right, so just to show you what this tree looks like when you make a tree with 1,500 species, just roll it across the floor here. You guys do it together. There you go. Don't pull it. Okay. All right. This is our magic scroll. Do not rip it because it took them forever to print this thing out. And it's going to keep going and more and further. There we go. So this is the largest phylogeny of land plants to date, or green plants to date, spread out in front of you on the floor. Very small font still, but 1,500 species. And this is an analysis of all of their genomes in it. So it's using this sort of divide and conquer approach where we think the computer's probably doing a pretty good job. How's that? To give us the answer. And so one way to look at it is here on the floor, spread out in front of us. Another way to look at it is in this, I call these hurricane phylogenies. I think they look like hurricanes. No one else does that, but these circular plots. And using all these other approaches, we know to infer across this big tree 245 ancient whole genome duplications across the history of green plants. And so, not only from analyzing the relationships of the species themselves, but all those duplication histories that are evident in each of those gene family phylogenies, we're able to see many, many duplications. And these are here indicated in these red and orange stars that are scattered throughout this tree. That's a lot. Before this study, we had about 60 that we know about. And so we really increased our knowledge and our placement as well of where these duplication events go in phylogeny. We have a naming convention for these. We call it the Tucson Naming Convention, since our lab is the leading lab for finding these in the world. And we call it that because we get the name. And so some folks want to call these like Norse gods and things, but there's not 245 Norse gods, as far as I know. So we just call them some kind of letter code with a Greek letter. How's that? To indicate this. So the most common one found in Arabidopsis, the first one was AT alpha, for example. Arabidopsis thaliana, alpha was the first, so, for example. They have some kind of naming system, because even though we got these dots on this tree, as we get additional species, right, this is still, even though it's 1,500 species, there are 400,000 plus or minus 50,000 species of green plants. So we don't necessarily know exactly where all of these go with that level of precision until we sample more species. But for many of these deep ones, we have an idea about where they are. So one of the fun things that comes out of analyzing data at this scale, and I hope that you can kind of see those plots, is that we can get a sense of how many times things have duplicated on average. So what we see here is, if we look at these violin plots here, you can see that we're going from the algae down here at the bottom all the way to the flowering plants here at the top. So angiosperms and gymnosperms and ferns and bryophytes and lycophytes and et cetera. What we see is that the numbers of rounds of whole genome duplication are they're non-randomly distributed across the tree of life of green plants. So flowering plants have many more rounds of polyploidy than the algae. We see little palliative polyploidy among the algae, almost none. And so going here from 0 on the x-axis to 6 on the right, 6 rounds. And so the way this is calculated is taking each of these tips, right, or the tips on this tree here, and working backwards to the root and saying how many times in history was each of these plants' genomes duplicated. And it turns out that if you were an algae, not very much. Almost none. There's very little. But if you're a flowering plant or a land plant, quite a few. And your average flowering plant genome has been duplicated 6 times in its history. So there's been 6 rounds of genome duplication. The average fern is about 6 times as well, or 4 to 6 times. Bryophytes and gymnosperms are a bit less, maybe 2 times they've had their experience to whole genome duplication. And the time frame we're talking about here for land plants is about the last 400 million years. And we, based on our statistical power analysis, we can see that far back in time with the approaches. So we're looking back at 500 million years of evolution. And we can see when and where these genome duplications happen, which I think is really amazing. So this gives us some perspective. So one of the fun things that we're testing right now is whether or not these genome duplications are associated with the evolution of multicellular. Most of these algal lineages are unicellular. Some of the ones, this came up because when we look at the lineages that do have polyploidy, it's always in the multicellular ones. And using our big machine learning process, we've been cranking through, I don't know, a couple thousand other data sets for other eukaryotes.